All right, hello everybody. In today's lecture, we are going to form sort of a bridge between the course that you're finishing now, which was focused mainly on metabolism, metabolic pathways, not exclusively, but quite a bit. And the next course, which is going to focus mainly on intercellular signaling and the various signaling molecules that cells exchange and the signaling pathways that are initiated by binding of these signaling molecules to receptors. So in this kind of a bridge lecture, I want to talk about unusual situation in cells. That's the name for the lecture. I think maybe in the schedule it still says extreme situations in cells, but that's probably not the best way of putting it. Because in lectures and seminars about metabolic pathways, usually we assume that the cells have sufficient amounts of substrates, sufficient amounts of oxygen, that they are doing fine, they're not doing anything special, they're just kind of chugging along. And today I want to focus on 3 situations where the cells have to do something other than that. They have to adapt to some changes. They have to adapt their metabolism, their functioning to some special states. And that requires some intracellular signaling because the cells need to figure out, they need to find out that they are in a different situation and they have to do something about it. And the 3 types of situations that I want to talk about, which are kind of special, even in normally functioning organisms like ours, many cells will encounter these special situations pretty much all the time, okay? So they sound like they're special, but they are more special with respect to the way that we discussed metabolism previously. But for many cells, they are kind of normal ways of functioning. And the 3 situations that I want to talk about are lack of nutrients, when the cells do not have enough substrates to actually to get enough ATP, basically. Lack of oxygen. Again, for some cells in our body, that's pretty normal to be at very, very low concentrations of oxygen. They're fine with that, but still, they need to know that they don't have enough oxygen. And the 3rd situation is kind of opposite to these 2, and that's a situation where the cell has everything that it needs in order to grow and proliferate. Again, that's something that the cell needs to figure out, needs to detect. Okay, I have enough signals, I have enough nutrients, I have well-functioning DNA. I can proliferate. Okay, so these 3 situations, special situations, are what we will cover in this lecture. Because as we'll see, cells will need to reprogram, rebuild their metabolic pathways and focus on some metabolic pathways and maybe downregulate other metabolic pathways in order to do the program to survive or to grow. Okay, does this make sense? Good. These 3 situations, as I said, require some sensors, some intracellular sensors that detect which state the cell is in. Okay? Is there a lack of ATP? Is there a lack of oxygen? Is there enough substrates, etc., etc.? So we will start with the first of such sensors, which is present basically in all our cells and is extremely important for the survival of cells. And this sensor, this signaling molecule inside our cells, is called AMP-dependent kinase. AMP standing for adenosine monophosphate. Now, I will write it kind of unusually with a dash. Normally it's written without the dash. I just put the dash in to show you that it's AMP-dependent kinase rather than AMP kinase. Many people will say, and from time to time I will make the mistake as well, I will call it AMP kinase. But what is a kinase, generally speaking? What does a kinase do? It adds phosphate from what? From ATP, okay? So it takes the terminal phosphate from ATP and puts it onto some other molecule. Now, if you say AMP kinase, it sounds like it puts a phosphate onto AMP. But that's not what AMP kinase does. AMP kinase is a protein kinase, so it phosphorylates a protein, okay? And it is sensitive, or it is activated by the binding of AMP to the kinase, okay? So it doesn't phosphorylate AMP, it binds AMP, which activates it, and then it acts as a protein kinase. Now, I mean, you've heard about protein kinases, and you will hear a lot more about them in the next course when we look at signaling. And they are one of the most common methods of intracellular signaling. So basically, there are loads of protein kinases which phosphorylate all sorts of different proteins and influence their activity. What other protein kinases you've already heard of in this course of protein? Sorry? Pyruvate kinase. Well, is that a protein kinase? Okay, which protein kinases? Okay.
Protein kinase A.
Yeah, sure, protein kinase A, absolutely. An important signaling kinase. Any other? When we spoke about muscle, we spoke about one protein kinase there. That's not a protein kinase, okay, that phosphorylates creatine, which is not a protein. So calcium what? Yes, there is a calcium-sensitive kinase that we spoke about in the when we spoke about muscle.
Light chain myosin kinase.
Correct. Yeah, myosin light chain kinase. That's a protein kinase which phosphorylates the myosin light chain in smooth muscle in order to initiate contraction. Okay, so these are all the protein kinases. There are hundreds, probably thousands of them in our cells that just continuously phosphorylate all sorts of proteins. So AMP-dependent kinase is a protein kinase which is activated by AMP. Now, why AMP? So AMP kinase is a sensor of low ATP. Okay, that's one of the three situations that we spoke about. Not enough nutrients, something is not working, the cell does not have enough ATP, and this is the sensor that will get activated. AMP-dependent kinase is activated by low ATP. Now, what is the connection between low ATP and AMP?
I'm guessing that when there's a higher ratio of AMP to ATP, this protein is activated.
That's correct. Okay, so when there is more AMP to whatever ATP, yeah, in a way, the protein will become activated. That's absolutely true. But my question is, what connects low ATP and high AMP?
When there is low ATP in the muscles, the muscle creates AMP instead of ADP.
How does it, okay, can you explain that a little bit more? How does that work?
It cleaves two bonds instead of one.
Okay, so what you're suggesting is that basically we have ATP, which in an ATPase, whatever, myosin, whatever, first gets turned to ADP. And what you're saying is that if there's not enough ATP, this ADP can be turned to AMP. Is that what you're saying? Or yeah, okay. So this is actually not what happens, or not directly. So basically, no ATPases are capable of using both ATP and then ADP. So they can't really cut away this additional phosphate in ADP. Okay? So most of the enzymes that use ATP, they can't really use ADP and just cut it out. I mean, it would make sense, right? Because the next phosphate is the same phosphate as the previous one. But that's not how it works. There's a reaction, and I'm pretty sure you've heard about it. There's a reaction that we can actually turn ADP to ATP.
ADP to ATP?
Mm-hmm. And not in ATP synthase, obviously. I mean, that's the obvious way, but yeah, I mean, so that's what we would do in ATP synthase, right? ATP synthase in oxidative phosphorylation would take ADP and make it into ATP. But that's not what I mean. Without using any additional energy, we can actually make ATP from ADP. No? How about making ATP from 2 molecules of ADP? No? Yeah? Okay, there's a process where our cells can take 2 molecules of ADP and basically shift one of the phosphates to the other one. And what we get is ATP plus AMP. And this is what happens in cells that have decreasing amounts of ATP, because ADP is not useful. No enzyme can actually use it for anything. So this reaction will occur where with using 2 molecules of ADP, we can make 1 molecule of ATP, which we can use for whatever we need, okay? And 1 molecule of AMP. Does anyone, now that you see the whole reaction, does anyone remember what the name of the enzyme is? It is a transferase. I will agree with that. It is a transferase. But what is the name of the transferase? It's called adenylate kinase. Adenylate kinase. Okay. It is adenylate kinase. And it is an absolutely essential enzyme in our cells, okay? Because it basically kind of doubles the amount of ATP that we can get out. It's not exactly doubles, but you know what I mean. Like we can keep using even the energy from the ADP to some extent, okay? Very, very important reaction. Now, this reaction is the connection between low ATP and high AMP, because at low ATP, this reaction will occur and we will get increasing amounts of AMP. Okay, so this is the mechanism. Make sense? So, this is what activates AMP-dependent kinase, and it is a signal for the cell. There's not enough ATP, we're running out of ATP, and we need to do something about it. The activation itself of AMP-dependent kinase is a tiny bit more complicated. It needs to bind AMP, and then some phosphorylations happen to it. That's not really super important. Okay, we don't need to go into such details. But the important thing is, what does the activated AMP kinase actually do? What kind of changes to metabolism by phosphorylating many different proteins? What kind of changes to the metabolism we can expect? So if you were to guess, okay, so the cell is running out of ATP. If you were the cell, what kind of metabolic pathways would you upregulate, and what kind of metabolic pathways would you downregulate in this situation?
Break down glycogen.
Okay, so one of the things you can start is glycogenolysis, okay? So breaking down of glycogen because we can get some glucose from the stores. Okay. That is true.
Gluconeogenesis also.
Gluconeogenesis, would you upregulate it or downregulate it?
Downregulated.
Why would you downregulate it?
Because you produce glucose and not ATP.
Okay, and moreover,
You would prefer to do glycolysis than gluconeogenesis.
I completely agree. So we would upregulate glycolysis because glycolysis will give us ATP directly and also indirectly in the Krebs cycle, right? So we would upregulate glycolysis, and we would probably downregulate gluconeogenesis because not only it doesn't produce ATP, it actually consumes ATP, right? Gluconeogenesis.
Muscles.
Huh?
Only the muscles will do it.
Yeah, but here,
Generally, you need to do gluconeogenesis to
What-
bring back glucose into the blood.
yes, but the trouble is, if you don't have enough ATP, you can't do gluconeogenesis because you don't have ATP. I see where you're coming from, okay? But here, we're looking at the context of an individual cell, not the whole organism. So for the whole organism, yes, it will signal, let's make more glucose because we don't have enough glucose. But in a cell, if the cell itself doesn't have enough ATP, it can't start making more glucose because it doesn't have enough ATP to actually do it, okay? So in the liver cell, if the liver cell has low amount of ATP, it will actually downregulate gluconeogenesis because it doesn't have enough ATP even to survive, let alone to make glucose for export, okay? So I see where you're coming from, but within a cell, actually, gluconeogenesis will be downregulated. What other pathways would you upregulate and downregulate? Beta oxidation, okay. Here I will just put it in a bracket because the effect is a little indirect, okay, but we could increase beta oxidation, that is true. Quite indirectly, but yeah.
You increase ketone metabolism.
What do you mean, increase ketone metabolism?
When you will run of glucose and you will have a lot of acetyl-CoA, you will prefer to do a ketone body metabolism,
Okay,
produce ATP indirectly to the brains and other organs.
sure. So what you're saying is that the cell could adapt to using ketone bodies because it is starving or whatever. Is that what you're saying? That you would like upregulate the uptake or the use of ketone bodies. Okay, again, I see what you're thinking about, okay, because in starvation, okay, we will produce ketone bodies, our liver will produce ketone bodies for the rest of the body. I get that, but here we are not talking, once again, about the whole organismal context, okay? We're not talking about signaling in starvation. We're looking at an individual cell, and the individual cell doesn't really know if there are ketone bodies around or not around, or what is around or not, okay? It only knows that it's lacking ATP and needs to do something about it, okay? So ketone bodies metabolism may potentially be influenced, but that's not going to be one of the main things to occur, okay? Now, connected to beta oxidation, one of the things that the cell can do, I mean, here we had glycogenolysis, right? So what other stores of energy can cells have? Fat, okay? So we will actually increase lipolysis. Not all cells will have lipid droplets, but if they do, actually AMP kinase will induce lipolysis so that fatty acids are produced and we get more substrates, okay? What other pathways, I mean, connected to glycolysis, in order to have glycolysis produce ATP, we need the substrate for that, right? Which is, wait, let's stay with glycolysis, okay? So for glycolysis, we need the substrate for glycolysis, which is, the substrate for glycolysis is glucose, okay? How do we get glucose into the cell? One possibility is from glycogenolysis. If it has any glycogen, that's fine. But if it doesn't have any glycogen, how do we get glucose into the cell? Correct. So we need to upregulate, or we need to put the transporters for glucose into the membrane. Otherwise, we don't get any glucose. So another thing that will be upregulated by AMP kinase are actually GLUT transporters. And one of the transporters, one of the subtypes of GLUT that will be upregulated by AMP-dependent kinase is GLUT4. Now, what do you know about GLUT4? That it's quite present in muscle and fat, I agree. And that it's insulin sensitive, okay? Thank you for saying insulin sensitive, not insulin dependent, okay? It's insulin sensitive. And what does it mean that it's insulin sensitive? It means that when insulin binds to a receptor, through a lot of steps, GLUT4 will be actually put into the membrane so that muscle, adipose tissue, and some other tissues can accept a lot of glucose. That is all true, that is absolutely true. But imagine a situation, okay? You woke up late, your 8 a.m. lecture is starting in whatever, 20 minutes, okay? And you actually want to go, okay? And you had to run to catch a bus, okay? So your muscles will require quite a lot of glucose. I mean, they will be using, of course, fatty acids, etc. But let's say you're really sprinting to catch the bus. So your muscles will need quite a bit of glucose, and they need to put transporters like GLUT4 into the membrane. But since you woke up and you had no breakfast, your insulin levels are going to be basically zero, okay? There's no insulin, or virtually no insulin, okay? So if the only signal for GLUT4 to get into the membrane was insulin, you couldn't run, right? You would have to eat something or whatever to get the insulin in. So what I'm trying to say is that for GLUT4, and also GLUT1, okay, another one of the possible signals to get them into the membrane is actually the activation of AMP-dependent kinase. Okay? Because the muscles, as they start working, they figure out, oh, I don't have enough ATP, okay? I need to start the pathways to produce ATP, and one of the important things is to get all the transporters in there, okay? So GLUT4, one of the signals for GLUT4 to get into the membrane is insulin, but the other one, or another one, is AMP-dependent kinase. There is actually a third signal, and I will just put it in brackets, okay? Calcium in the muscle will also induce the translocation of GLUT4 into the membrane, okay? It makes sense, right? Calcium means contraction, okay? So it's a similar signal just telling the muscle, let's put GLUT4 into the membrane, okay? So this is just to show you that GLUT4 will respond to insulin, but it will also respond to all sorts of other signals. So we have transporters which need to be put into the membrane. What other pathways will be stimulated or downregulated in this situation? Not enough ATP. What about pentose phosphate pathway? Why would you upregulate pentose phosphate pathway?
Because it gives you the glycerol phosphate.
It gives you...
Then
Okay, I see what you're saying. Yes, in the end, pentose phosphate pathway could actually produce indirectly ATP, but pentose phosphate pathway's primary purpose, or two primary purposes, are NADPH, which is needed for antioxidation. Sure, sure, that's true. For lipogenesis and all sorts of other things, for actually building stuff. Okay, NADPH is really used for building stuff. And the other purpose for pentose phosphate pathway, in addition to NADPH? Ribose for DNA synthesis. So the primary purpose of pentose phosphate pathway is actually to get building blocks if the cell needs to grow and if it needs to synthesize things. So pentose phosphate pathway will not be upregulated by AMP kinase, AMP-dependent kinase, because that's actually a situation where you don't want to start building stuff because you don't have enough ATP, okay? So not really. I see how you mean that, okay? But pentose phosphate pathway will not be upregulated.
But will it be downregulated?
It will be probably indirectly downregulated by shifting most of the glucose into glycolysis, okay? So there will not be enough substrate left for pentose phosphate pathway. Huh? What about it?
Downregulated?
Yeah, it will probably be downregulated. I'm not 100% sure whether through AMP-dependent kinase or through other processes. But you're right. But urea cycle is really present only in one tissue, right? Which is the liver, right? It really only occurs in the liver. So that's quite a specific thing. But in the liver, it will be downregulated. I'm not 100% sure whether through this mechanism, but it will be downregulated. So good thinking, yeah, because it's a highly ATP consumptive process, okay? There's still a few pathways that will be upregulated or downregulated. Proteolysis. Okay, again, that would be probably more specific to skeletal muscle because there's a lot of protein there. You don't really want to break down proteins in other tissues because that could be a problem. But you're quite actually close to, we could call it proteolysis, but which is all connected together. The thing that will be upregulated is called autophagy. I'll explain what that means. Autophagy means eating oneself, right? And that's a very, very important process in our cells that are struggling with the availability of various substrates, where they actually start breaking down their organelles into the individual building blocks, lipids, protein, amino acids, etc., and using them for either energy production or for building something that they absolutely need. So one of the things AMP kinase will really upregulate is this process for autophagy. You will probably hear about autophagy quite a lot. It is a process that is now becoming very fashionable in various diseases and figuring out what goes on in aging and slowing down aging, etc. So autophagy is very close to what you're saying. So it's not going to be a large-scale proteolysis like in a muscle, in a skeletal muscle. But there is definitely upregulation of autophagy just to get scavenge, lipids or bits of mitochondria that you could use to make ATP. I agree. Okay. Now, related to all these very, very basic pathways, we still haven't really touched on another pathway which is very useful to make ATP. The Krebs cycle, right? The Krebs cycle. So we have glycolysis. Yeah, absolutely. That's great. Okay. But if we can use it, Krebs cycle is much better, right? Because we get a lot more ATP. So the Krebs cycle will absolutely be upregulated. Absolutely. And I will show you in a second what are the specific targets that are being modulated by AMP-dependent kinase. Going back to the pathways that you want to downregulate, okay, and start very simple. What are the things that you absolutely do not want to be doing? So pentose phosphate pathway would be one of them, potentially. Huh?
Glyconeogenesis.
Okay, so glycogen synthesis, yeah, absolutely, I agree. Yeah, that will be downregulated, I agree. What else? Lipogenesis, so fatty acid synthesis, right? You absolutely don't want to do that, yeah? What else? What else uses a lot of ATP and, you can't really do it? Closely related to fatty acid synthesis or ketone synthesis, ketone body synthesis. What else is a really big molecule that takes a lot of ATP to build? Cholesterol, absolutely. You would downregulate cholesterol synthesis. And for most cells, one of the most ATP-consuming processes is, and I'm aware that you probably never heard it, but what do you think is, in most cells, is the biggest consumer of ATP? Which process? Apoptosis. Well, potentially, but that's the end, okay? But for normally functioning cells, what will they spend most of their ATP for? Protein synthesis, absolutely, protein synthesis. So we would downregulate protein synthesis because it uses a lot of ATP, okay? And I will show you in a second how that is mediated. All right, I think that we've covered all the things that I've written down. Now, we can now have a look at what are the specific targets, okay? For glycogenolysis, the most likely target is the glycogen phosphorylase kinase, okay? So this is the regulation of glycogen phosphorylase. For glycolysis, the main target is phosphofructokinase 2. That's the one that makes fructose. That would be phosphofructokinase 1.
Can you repeat that?
Oh, 2,6.
Okay, yeah, that's so the question was, what does phosphofructokinase 2 do, okay? And that makes, say it again, 2,6-bisphosphate, okay, fructose 2,6-bisphosphate, which is an allosteric regulator, which then activates glycolysis, okay? So kinase 2 is the stuff which is actually phosphorylated by AMP-dependent kinase. With beta oxidation, as I said, that's a little bit indirect. With lipolysis, there is an enzyme called ATGL, which means adipose tissue triglyceride lipase, okay, which is basically just taking care of the lipid droplets in there. Okay, so that's the one which will be regulated. With the glutes, it's not phosphorylating the glutes themselves, but actually proteins which push them into the membrane. Okay, I will not go into details. Autophagy, a little bit complicated. We don't really need to know that. For Krebs cycle, the enzyme which will be targeted is pyruvate dehydrogenase. Okay, that's the gatekeeper enzyme. So PDH is affected by AMP kinase. For gluconeogenesis, how do we downregulate gluconeogenesis? We don't know, okay? It does occur, but it's not really known. There are a few hypotheses, but it's still unclear. Huh? No. Again, we're talking about what happens in the cell, okay? The cell itself. So glucagon is an additional signal which signals something and which does something, okay? But here we're just talking within the individual cell. How does it influence? How does AMP kinase influence gluconeogenesis? There are a few hypotheses through some transcription factors, etc., but it's not clear how that actually works. But it does happen, okay? You will hear about the decrease in gluconeogenesis once you start in year 4 or whatever, once you start learning about the treatment of diabetes. One of the drugs which is used to treat diabetes is called metformin, very widely used. One of the effects of metformins is activation of AMP-dependent kinase, and we know that metformin decreases gluconeogenesis in the liver. It's only that we're not sure what the steps are between those two, okay? So that's the reason why I'm also teaching this. Fatty acid synthesis is downregulated by or decreasing the activity of the first enzyme of the pathway, which is fatty acid synthesis. Acetyl-CoA's carboxylase, okay? So the formation of malonyl-CoA, okay? So that's the enzyme which is whose activity is downgraded or downregulated by AMP-dependent kinase. For cholesterol, again, we have the main rate-limiting step, which is exactly AMG-CoA reductase. So that's the one that's influenced by it. And for protein synthesis, this is actually a bit, again, indirect, but I will now tell you what this indirect pathway is, because that's the next part of the lecture. Because AMP-dependent kinase decreases the activity of another sensor in the cell, which is then responsible for increasing protein synthesis. I'll say that again. AMP-dependent kinase will decrease the activity of the next sensor that we'll talk about, which upregulates protein synthesis, and that is called mTOR complex. Okay, so mTOR complex, and we will spend quite a bit of time with mTOR complex, among other things, increases protein synthesis, and AMP-dependent kinase decreases the activity of mTOR complex, therefore it decreases protein synthesis. Okay? All right. Any questions about AMP-dependent kinase? If there are none, I will just repeat, okay? The meaning of the activation of AMP-dependent kinase is we don't have enough ATP. So what we need to do is upregulate everything that makes ATP and downregulate everything that consumes ATP. Okay, and these are the steps that our cells will do by using the sensor. All right, the next sensor. Can I erase it? Yeah. The next sensor is this mTOR complex. What does the abbreviation stand for? What is an mTOR complex? Well, first of all, what is an mTOR complex? mTOR complex is a massive protein complex. In its dimeric form, because it forms dimers, in its dimeric form, it weighs over 1 megadalton, 1 million daltons. It's a huge protein composed of many different subunits, okay? Really, really big protein. The name, we can break it down. The name starts with M, which originally stood for mammalian, because it is present in mammalian cells. I mean, now they say that it's mechanistic, but whatever. Mammalian, I think that makes a lot more sense. The TOR part stands for target of rapamycin, and I'll explain what that means. Rapamycin is an antibiotic. It's a fairly large molecule produced by a fungus. And it was first discovered in the 1960s, I think, in the Easter Island. And the Easter Islands are called by the natives there Rapa Nui. That's the name for the Easter Island, Rapa Nui. Hence the name rapamycin, because it was found in the soil there. Okay, so I'll say it again. Rapamycin is an antibiotic produced by some fungus, Streptomyces hygroscopicus, or something like that. And since it was found in Rapa Nui, they called it rapamycin. Now, for a long time, it had no use because it's not a very good antimicrobial agent, and because most antibiotics are used as to treat infections, right? And also, it had quite a lot of side effects, so basically, it wasn't really used for a long time. I think there's a story that one of the discoverers, or one of the people who were working on rapamycin, they were working for some pharmaceutical company, and the pharma company had no interest in this drug. So the guy just basically stole samples of rapamycin and started researching them for other things. So there's quite an interesting story. But anyway, rapamycin was found to have a really, really good property or effect, and that is it can work as an immunosuppressant. It can decrease immune reaction in humans, and this is the main use nowadays. Okay, it's not one of the main ones, but it does that. Now, researchers started looking at what is the target of rapamycin, what does it bind to in our cells in order to produce this immunosuppressant effect. And first, they found a target, a binding protein in yeast, I think, and they called it target rapamycin, okay? And then they find a homologue in humans, or in mammals, maybe it was in mice or something like that, and they call it mammalian target of rapamycin, okay? So hence the name. So rapamycin binds to this mTOR complex, and the C stands for complex because it's huge, okay? So rapamycin binds to this complex and influences its activity, but that's not what mTOR complex is for, right? As you would expect, it's just a kind of a random effect which occurs. So what is the purpose of mTOR complex? mTOR complex is a signaling sensor which, when activated, tells the cell that it has everything that it needs in order to grow and proliferate. Okay? So when mTOR complex becomes activated, the cell can start to grow and proliferate. If it's not, the cell will not do this. Okay? So it's a switch between proliferate, don't proliferate. Now, mTOR complex, as I said, is massive. It's really, really huge. And it integrates together quite a lot of different signals. So you can actually think about it as a tiny computer which detects all sorts of different things in the cell. And if everything is correct, so it will basically, you can imagine it as a checklist. And if everything is correct, then it will give the signal, okay, now you can start producing proteins, you can divide DNA, et cetera, et cetera. What are the main signals that the mTOR complex actually detects in order to give this signal that we have plenty of stuff? One of them is the availability of amino acids. And the main amino acids that the mTOR complex detects, again, through fairly complicated mechanisms, are essential amino acids. And among them, the most important one are leucine and arginine. Okay? Some of the other ones can also have an effect, but the availability of leucine and arginine are the main signals for mTOR complex that the cell has enough. Okay, that it actually has enough amino acids in order to do stuff with them. Okay, so amino acids are signal number one. Signal number two is basically external stimulation for the cell which tells it to grow and proliferate. So this usually is in the form of growth factors. And you will hear about growth factors quite a bit in the next course when we spoke about all sorts of signals that tell cells to grow. So this is another signal. Okay, from the outside, we are getting all the signals to grow. Let's grow. The next signal is that we have enough ATP, right? We need to have enough ATP in order to be able to grow. Now, what is the signal for sufficient amounts of ATP? Or lack thereof? AMP-dependent kinase, right? So basically, if AMP-dependent kinase is activated, this signal is not going to be present. It's going to be a negative signal. And if AMP kinase is not activated, then mTOR complex will assume that we have plenty of ATP. Does it make sense? So the next signal is availability of ATP, but it kind of works the other way. So if there's not enough ATP, AMP-dependent kinase will downregulate, will decrease the activity of mTOR complex. Makes sense? Okay. The fourth signal that I want you to know about is the quality, the integrity of DNA. Okay? So the next signal is our DNA is okay, we can start growing. Okay? It's not damaged or anything like that. So I'll put in DNA okay. In other words, if the DNA is damaged, mTOR complex will be downregulated, rather the activity will be decreased, and the cell will not have an incentive to grow because the DNA is damaged, okay? I will not go through the specific steps, how DNA damage is communicated to mTOR complex. That's not important at this level. Just be aware that it's another important signal for mTOR complex. Now, as I said, the signaling on mTOR complex is very, very complicated, and it's still not completely understood. So there are also other signals that probably play a role. But in this simplified form, if all those things are met, if all those conditions are met, then the cell will start dividing and will start building proteins, DNA, etc., etc. Okay, that's what it says. Now, why am I spending so much time with mTOR complex? So the same way AMP-dependent kinase is important for medicine because metformin, the anti-diabetic drug, works indirectly through AMP-dependent kinase. The mTOR complex is absolutely crucial for cancer cells to grow and to divide and to get enough signal. Basically, if mTOR complex is blocked or downregulated in many cancer cells will stop dividing. They don't have enough signal. Okay. Now we can't easily use rapamycin for that because things are a little more complicated. Okay. But for the growth of normal cells and cancer cells, mTOR complex is absolutely crucial. Now, since mTOR complex is the signal of plenty, we have enough, we can start growing, what would be the pathways, what would be the things that mTOR complex could activate, and what are the things that mTOR complex could deactivate? So we spoke about some of them already. So
It will activate protein synthesis.
absolutely, it will activate protein synthesis. Right? And DNA synthesis, absolutely, including the synthesis of nucleotides. Okay, so not just DNA synthesis as in replication of DNA, but the synthesis of nucleotides, so the building blocks of nucleic acids. Absolutely. What else could be?
Krebs cycle.
The Krebs cycle, okay. Potentially, but just be aware that in order for the mTOR complex to be activated, we need to have enough ATP. Okay? So if you don't have enough ATP, this thing will not be switched on. So the mTOR complex more or less assumes that ATP is cared for. Okay? So there's not going to be a lot of effects on the normal energy metabolism because that is assumed to be okay because we have enough ATP. Okay? So this is not going to be one of the main ones.
Gluconeogenesis and pentose phosphate.
The pentose phosphate pathway, absolutely, yeah. The pentose phosphate pathway will be upregulated because we need to make nucleotides, okay? Now this will be probably a little bit indirectly through the mTOR complex, but yes. So it will not be directly, but there will be some indirect effects. But again, gluconeogenesis is irrelevant for the cell itself. Gluconeogenesis is only relevant if we look at the organism as a whole, okay? But if a cell needs to grow, it will not start producing glucose for itself. It doesn't make any sense, right? Why would it do it? It's just a waste of energy because then you would have to take the glucose and break it down again, right? So gluconeogenesis really works only, okay, the whole organism is starving, we have some protein or whatever, let's make glucose for the organs that need it. But in the context of an individual cell, it doesn't really make any sense to make glucose.
It's only unique in the liver.
Absolutely, it only works in the liver, okay? And it needs very specific signals which are not in the cell, but are actually coming from the outside. So you have to make the cell make glucose because for the cell itself, it doesn't make any sense. It's just using up their energy. Does that make sense? Okay, so gluconeogenesis is probably not going to be the main one. We need to grow the cell. We need to make some building blocks, right? So we are making proteins, we're making nucleotides. What else do we need in order to grow a cell? Lipids in general, right? We need fatty acids, we need phospholipids. Actually, a lot of the cell are just membranes. We need to make a lot of lipids, okay? So lipid synthesis, including cholesterol synthesis, will be upregulated, okay? Because the cell needs to grow. With respect, I will go back to how you suggested that Krebs cycle might be upregulated. In fact, one of the things that mTOR complex, again, indirectly will activate is actually glycolysis. And I will explain towards the end of the lecture why glycolysis is preferable. If the cell wants to grow, it will actually generally prefer glycolysis to Krebs cycle and to complete oxidation of glucose, okay? Just remember that this is a weird thing, okay? And I will explain that towards the end of the lecture, why that is actually useful. So glycolysis will be upregulated by the mTOR complex. The last thing that will be upregulated, or the last thing that I want to mention, is something that you may not have heard about, and that is mitogenesis, which is basically just production of mitochondria. Okay, so it will, on one hand, it will activate glycolysis, okay, to get a high throughput, a lot of glucose to pull through. On the other hand, it will actually make more mitochondria. Okay, again, it makes sense, right? The cell wants to divide, it has to make more mitochondria just in order to have enough mitochondria to divide between the daughter cells, right? Makes sense. One of the things that will be downregulated by mTOR complex, again, hopefully it makes sense, is autophagy, right? You don't want to be building new bits of the cell and at the same time digest them and break them down, right? So you would downregulate autophagy. Yes?
May I ask a question?
Yeah.
What does it mean, autophagy? Like you digest organelles?
Absolutely.
Whole cell?
No, just organelles. So it is autophagy means it's still within one cell, but basically what you get in the cell is, I don't know, you have a mitochondrion here, and the lysosomes will either just kind of bite a bit of it and digest it, or engulf the whole mitochondrion and digest it, or bits of endoplasmic reticulum, for example. Okay.
Which organelles you would prefer to digest?
Often it is mitochondria, which is then called mitophagy. Okay. It could be endoplasmic reticulum and Golgi apparatus, so basically the vesicles, the lipid-bound vesicles that will be digested. Those are the usual ones that will be digested by autophagy. Okay. Could be potentially other ones, but these are the ones, the main ones.
Autophagy of the nucleus?
Yeah, I don't think that happens. I don't think that happens. You don't really want to break the nucleus because that will cause problems.
Isn't it apoptosis when you digest the nucleus?
Yes, but that's not done by autophagy. So autophagy is a highly controlled process where you just kind of, the lysosome will engulf a part of the organelle and destroy it and degrade it and digest it, basically. With the nucleus in apoptosis, you just kind of start breaking down the DNA and everything, but using a different mechanism. Okay, so that's not autophagy, that's apoptosis. Good. So this is the mTOR complex. Now, I suggest we take a short break, and then we'll cover the last sensor, which is going to be the sensor of hypoxia. So let's meet again in 5 minutes. Yep. All right, so moving on. This is going to be the third and last sensor that we will talk about. And as I said, as I foreshadowed, it's going to be the sensor of hypoxia. So not enough oxygen in the cell. And as you can imagine, that is, for many cells, that is a big problem, right? Not enough oxygen, it means we can't use some metabolic pathways. And the cell needs to adapt, needs to react, usually very quickly. Now, in terms of pathology, one of the common situations where the cells suddenly do not have enough oxygen is caused by what's called ischemia. Have you heard of ischemia? Does anyone know? Okay, ischemia basically means a state of not enough supply of oxygen, not enough supply of nutrients. Oftentimes it is caused by a blocked blood vessel, right? Either in the heart or in the brain or elsewhere, and it causes acute ischemia of the cells, which means not enough nutrients, not enough oxygen, and they need to do something about it. And one of the sensors which will be activated, is the one that we saw as the first one. AMP-dependent kinase will absolutely be activated in this situation because we're not making any ATP, right? Without oxygen, without nutrients, we can't make ATP, so AMP kinase will absolutely be activated. The other sensor, as I said, is specific to hypoxia, because of course, lack of ATP can have other reasons, other causes, but the specific sensor for hypoxia is called hypoxia-inducible factor, or HIF, hypoxia-inducible factor. Now, how does it work? And this is actually one of the cleverest tricks of evolution, because imagine that you have to make a sensor which will start working in a situation where the cell basically can't do very much, right? No oxygen, you know, your ATP stores are probably going down. You have to do something. But if you make a sensor which has to produce something or do something, usually you need ATP for that or something, something complicated. Here it's quite interesting. So the HIF protein itself can be said that it has two subunits. I will explain, it will become clearer how that works with subunits. So we have HIF alpha and HIF beta. I will mention HIF beta later on. But HIF alpha is a protein which sits in the cytoplasm, or is being produced in the cytoplasm, let's put it that way. And in the presence of oxygen, it is being continuously broken down. Say it again, okay? So HIF is being produced in ribosomes, and basically as soon as it is produced, it's broken down. But only if there is enough oxygen. If there is a sudden lack of oxygen, this HIF which is produced is not going to get broken down, and it can start signaling. Okay? So it's a sort of a switch which basically starts working by something not working. Hope that makes sense. Okay? So I'll give you more details about that. So if there is enough oxygen present, HIF1 alpha, or HIF alpha, sorry, there are three types of HIF, 1, 2, and 3. We don't really need to deal with the differences very much, okay? So I'll just call them HIF, but there are some subtypes of HIF. HIF alpha will get broken down in the presence of oxygen by means of, or the process is started by enzymes called prolyl hydroxylases. And I think you've probably heard of prolyl hydroxylases in some context. Yeah, and more specifically, which part of the bone? Which component of the bone? Okay, collagen, right? In collagen, we hydroxylate proline, okay? So this is a different situation, but we also have prolyl hydroxylases here. And these prolyl hydroxylases basically, so if we have the protein and there are some prolines in there, these prolyl hydroxylases will put OH groups on them. And for that, in order to hydroxylate these prolines in HIF alpha, they need oxygen, they need iron, non-heme iron, okay, so it's not iron in heme, but it's actually just the iron. And they also need, as a donor of electrons, they need 2-oxoglutarate. Okay, unusual, yeah, I agree, okay, but that's how it works, okay? So 2-oxoglutarate will just give electrons to this process, okay? And the oxygen will be used to hydroxylate proline. And the iron there is a catalyst for this thing to work. If you're interested in more details, you can probably look up how this works, okay? Succinate is produced from 2-ketoglutarate, so there is a decarboxylation and then succinate being produced, okay? But the 2-ketoglutarate 2-oxoglutarate just works as a donor of electrons for this thing to work. In other words, if any of those components are missing, the prolines will not be hydroxylated, and HIF alpha will not get destroyed because it gets destroyed by binding another protein, which is called von Hippel-Lindau protein. Has anyone heard of von Hippel-Lindau syndrome? The geneticists sometimes like to talk about it. It's a relatively rare syndrome where people start developing many tumors, some of them benign and some of them malignant. And it is caused by mutation in this specific protein, which is needed to degrade HIF alpha. Okay? So what I'm just saying is that if you kind of start, if you can't degrade HIF alpha at normal oxygen levels, you will start developing tumors. Okay? So that's called von Hippel-Lindau syndrome. So the von Hippel-Lindau protein binds to this hydroxylated HIF alpha and uses ubiquitin to label it, and then the whole thing is then degraded in the proteasome. Okay? So it becomes ubiquinated, ubiquitin, and degraded in proteasome. So once again, if there is enough oxygen and there is enough 2-ketoglutarate and iron, HIF-alpha gets hydroxylated on the proline residues by prolyl hydroxylases. These hydroxylated prolines will bind von Hippel-Lindau protein. Just to give you the spelling, von Hippel-Lindau, which then causes the ubiquitination of the HIF-alpha and destruction in proteasome. Okay, so that's what's happening in our cells when they have enough oxygen. If there is not enough oxygen, or potentially not enough iron, or not enough 2-ketoglutarate, or too much of something else, but I'll leave those possibilities aside. So if there's not enough oxygen, the hydroxylation doesn't occur, and the HIF-alpha gets translocated into the nucleus, where it binds to the DNA with its partner HIF-beta. So we have HIF-alpha, which dimerizes with HIF-beta, and they work as a transcription factor. So not enough oxygen, HIF is being stabilized, translocates into the nucleus, and forms a dimer with HIF-beta, and starts influencing the transcription of various genes. Just by this, you can already see that the signaling through HIF is much slower than the signaling through, for example, AMP kinase, AMP-dependent kinase, right? AMP-dependent kinase will be activated in probably fraction of a second and will start doing things. Here, the stuff is a lot more complicated. So first, you have to stabilize HIF, you have to translocate it into nucleus, you have to start producing RNA that needs to be then translated into proteins. So it's a much, much slower signaling. But it is still quite important because it allows many cells to adapt in long term to lack of oxygen so that they can survive. Okay? So it's not going to be the acute reaction, but it's going to be nevertheless an important reaction to a lack of oxygen. So what do you think could be the pathways or the enzymes that could be upregulated? Because we're now talking about transcription, right? So we're literally making more of the proteins. We're not influencing their activity. We're making more of them. So what would be the things you would make more of if you don't have enough oxygen? Huh? Sure, that is true. Once again, we're now mostly thinking about the context of one cell, okay? But you're right. Actually, one of the genes that will be upregulated in some cells will be the genes for signals that make either more red blood cells or that may more blood vessels. So I will agree, okay? One of the things that will be upregulated are the genes for erythropoietin, which is the hormone which activates the production of red blood cells. But only very few cells produce erythropoietin, okay? Mostly they are in the kidneys. There are tubular cells in the kidneys. Those are the ones that produce most of erythropoietin. Normal cells will not really produce erythropoietin. So this is quite specific to just tubular cells in the kidneys. But there's another signal which can be secreted by many cells, and that is called VEGF, which stands for vascular endothelial growth factor. And that is a growth factor which tells endothelial cells to start growing towards the cells so that new blood vessels are formed and there's more oxygen coming in. And this is one of the mechanisms through which, for example, cancer cells are capable to basically make the surroundings to bring more nutrients and oxygen to them. So they will secrete this VEGF through this signaling or through some other signals, but that's one possibility, okay, to basically make new blood vessels to grow into the tumor. Okay, this process is called neoangiogenesis. And it is one of the targets for cancer treatment these days. So because we have antibodies against VEGF, so we can block this growth of new blood vessels into the tumor. And yeah, we can help decrease the size of the tumor or the rate of growth. So this is one possibility, I agree, okay? But within the cell, right? So not enough oxygen, what do we do? Huh? Absolutely, we would upregulate glycolysis. And since, in this case, we are absolutely sure that it's going to be anaerobic glycolysis because we don't have enough oxygen. Yeah, absolutely. So we'll also upregulate lactate dehydrogenase, which is the necessary enzyme in order to make lactate and to have the anaerobic glycolysis to work. Okay? So including LDH. I think the main enzymes are LDH and hexokinase, which are being stimulated by this. Once again, because we need to increase glycolysis, we also need to get enough glucose in, right? So we would also upregulate the amounts of GLUT transporters. I think in this case it's mostly 1 and 3 which are upregulated by the signaling, but the logic is the same, right? We need glycolysis to work, we need glucose to get in. All right, what are the things you would downregulate if you don't have enough oxygen? Which things are not going to work? Absolutely, we would downregulate electron transport chain. I think the main effect is through the downregulation of the amount of complex 4. That's the one that reacts with oxygen, right? So complex 4 is the main one, but we just decrease the amount of the electron transport chain because it's not really useful very much without oxygen, right? I agree. Connected to the electron transport chain is the Krebs cycle. Yep. So downregulate the Krebs cycle, mainly through PDH, through pyruvate dehydrogenase, the gatekeeping enzyme for the Krebs cycle. Okay, so we would decrease the amount of PDH because the Krebs cycle can't really work without oxygen. The last thing that you would downregulate is the formation, the growth of new mitochondria. Again, you know, we can't really use mitochondria without oxygen. I mean, we can use them a little bit, but not very much. So we would also decrease mitogenesis. If you're interested, because I mentioned mitogenesis a few times already, the growth of new mitochondria, the main pathway, signaling pathway for the formation, for the stimulation of growth of mitochondria is a transcription factor called PGC1 alpha. It's not super important that you know it, but maybe if you just hear it, it may come up again. PGC1 alpha is kind of the master regulator of making new mitochondria. Okay, so if the cell needs to grow more mitochondria, this is the transcription factor that will be activated, and it will activate the whole program of making more mitochondria. All right, any questions about HIF and the type of signaling? Okay, I would just say, because I mentioned that there are 3 different subtypes of HIF, 1, 2, 3. Okay, one is the one that most cells have, and that is responsible for regulating glycolysis and kind of the metabolic things. HIF2 is more responsible for upregulating these signals for making either red blood cells or growing new blood vessels, okay? And HIF3, I don't think it's very clear what that one does, okay? So there are some differences between the different subtypes. Not necessary to know that, but just be aware that there are some functional differences. All right. If there are no questions about HIF signaling, in the last part of the lecture, I just want to walk you through a few situations, mostly pathological situations, and to think together with you how the cell will try to adapt, what kind of sensors will be switched on, and what will happen. So the first situation is just hypoxia. And I already mentioned ischemia, which is a combination of hypoxia and lack of nutrients, and we will get to that. But let's now think about pure hypoxia. What kind of situation would you expect the cell to have just pure hypoxia? So plenty of nutrients, but not enough oxygen. What kind of situation would a human body, human cell find itself in hypoxia? High altitude, yeah, absolutely. That's probably the most common, right? I mean, it could be in an airplane because there is actually lower pressure of oxygen. Yeah, that's a good example of hypoxia. So what will be happening in a cell if it is faced with hypoxia? What are the sensors that will be activated? Which sensors will be deactivated out of the three that we talked about? And what metabolic pathways will be upregulated and downregulated? I mean, this one should be relatively straightforward. Absolutely. So HIF is activated. Well, it's always the alpha subunit, which is stabilized. The beta subunit basically sits in the nucleus and just forms a dimer. So the beta is just there all the time, okay? So it's always going to be the alpha subunit. Whether it's going to be HIF 1 or 2 or 3, that depends on the cell, etc. Okay? But yeah, HIF will be activated. Now, as I said, the signaling through HIF is relatively slow. It will take at least minutes, probably hours, to actually have an effect. Okay? So that's absolutely true. It will be activated. But what about the other ones? And why will AMP-dependent kinase be activated? Yeah, so for most cells rely on oxidative metabolism to produce enough ATP. So it's very likely that for many cells, when there's not enough oxygen, their ATP levels will go down. It's not necessarily true, right? There are some cell types that don't really use oxidative metabolism, so they will not even notice when there's not enough oxygen, but most cells will, okay? So yes, I agree that AMP kinase will likely be activated. What about mTOR complex? How is it going to be deactivated in hypoxia? Why? What will deactivate it? Correct, absolutely. So mTOR complex will be, its activity will be deactivated or decreased through the signaling of AMP kinase. So not directly, but indirectly through AMP kinase, right? Absolutely. So what will this cell, this hypothetical hypoxic cell, what will it try to do to survive? What kind of metabolic pathways or other processes will be upregulated or started?
The phagocytosis.
Autophagy. Sure, but again, autophagy is a relatively slow process. That's one thing. And the other thing is, most of the stuff that you get from autophagy is what? What kind of chemicals do you get when you start eating bits of the cell? You'll get proteins, amino acids, and fatty acids. Is that a good source of energy if you have hypoxia? Why not? Correct. All these things, with some exceptions, you need actually oxygen to metabolize, right? So autophagy will not be hugely useful in the case of hypoxia because most of the components will not really give you any ATP without oxygen, okay? So autophagy is not going to be probably the main thing that will be activated. Again, I am simplifying quite a lot, okay? So of course there are other signals that will be coming in that will actually block autophagy. So just looking at that, you would say, yeah, of course, autophagy will be activated. In this case, it probably will not be activated because there will be other signals telling it not to happen. But yeah, I see your thinking, but it will not be very useful for the cell to start eating itself in the case of hypoxia. So what will be useful? Glycolysis, absolutely, okay? So we'll upregulate glycolysis. And all the things that we need for glycolysis. So GLUT transporters, right, into the membrane to get as much glucose as we can, because in this case, it's absolutely going to be anaerobic glycolysis. We don't have enough oxygen, right? So we will not get a lot of ATP per molecule of glucose. That's why we just need to push a lot of it through. Otherwise, the cell is not going to survive, right? And at the same time, we will downregulate all the processes that are not necessary at the time. Protein synthesis, lipid synthesis, fatty acid synthesis, cholesterol synthesis, DNA synthesis, all those things will be downregulated, mainly through AMP kinase, AMP-dependent kinase, and mTOR complex, which is downregulated. Okay, so all these things will be repressed because it will be stupid to start making these components. Good. What about a different situation? It's a situation where we have a lack of nutrients, okay? We have plenty of oxygen, but there's a lack of nutrients. So I'll call it starvation, okay? But let's think about cellular starvation, not at the level of the whole organism, because then we have lots of other signals, but just the cell is starving, okay? what signals will be upregulated, which ones will be downregulated, and what will the cell try to do? Absolutely. Will be activated. Okay, so HIF is the standard default. It's not there, basically. It's destroyed. Yeah, I agree. So HIF is not there, okay? And mTOR complex is downregulated, is deactivated, right? Mainly through the action of AMP kinase, but not just that, because you know that mTOR complex directly senses the amount of nutrients, mainly leucine and arginine, so it already knows that there is not enough nutrients. What can the cell do in this situation? There are not enough nutrients. What will it do? Autophagy. Here it makes a lot of sense, right? We have plenty of oxygen, we're just lacking nutrients. So autophagy will be a very, very useful thing. What else could the cell do? Not enough nutrients, but it still needs to survive. So autophagy is definitely a good answer. What else could it do? Absolutely, so we'll be using its stores, if there are any stores, yeah. So glycogen and lipids will be used. What about it? Yeah, but once again, okay, for the cell itself, which is starving, gluconeogenesis is absolutely no help. It will not help the cell, okay? In the context of an organism, we can make the liver start making glucose because it can help the brain, etc. But for the cell itself, gluconeogenesis is actually, waste of energy because it's just making glucose for export and doesn't really see anything of it, okay? So if a cell is starving, it's not going to start producing glucose. Again, for the whole organism, it's a little bit different, okay? Because we can make the liver make glucose. But the cell itself is not going to make glucose. So gluconeogenesis will actually be downregulated. So your colleague already mentioned that, okay? So yeah, if we have some stores, they will be used. Glycogen, lipids, as in fat droplets will be used, absolutely. So glycogenolysis and lipolysis will be activated. I agree. Okay, but that actually only happens in like 2 types of cells, right? The first step in the muscle and the second step in the liver. It's very specialized, okay? Here we're mostly talking just about a normal cell, okay? Well, one thing I would suggest is the upregulation of the Krebs cycle. Why do you think I think that Krebs cycle could be upregulated? Okay, we don't have enough nutrients. Well, we might want to upregulate the Krebs cycle. Why do you think I think that? Yeah, but I mean, you're probably trying to focus on ATP production because you don't have a lot of it, okay? So potentially, but in hypoxia, you absolutely want to rely on anaerobic glycolysis. That's the only thing you have. But in a lack of nutrients, you probably don't, right? You do have enough oxygen.
If you're degrading lipids and proteins from the autophagy, you will receive a lot of acetyl-CoA. That will help you in Krebs cycle.
Absolutely. So one thing is that you're, breaking down lipids and amino acids, and for that you need the Krebs cycle. But also for glucose, if you use the Krebs cycle, you get a lot more ATP. It's a lot more efficient, right? So you would upregulate the Krebs cycle because that's what makes a lot of ATP for you, okay? So both the Krebs cycle and the electron transport chain will be upregulated and will be kept up because those are very, very good at making a lot of ATP, even with few nutrients. Okay. If we take those 2 situations together, we get ischemia. Ischemia, blood cell is blocked. We don't get enough oxygen. We don't get enough nutrients. So basically, in a heart attack or in a stroke, those cells are experiencing these two situations at the same time. Not enough ATP, not enough oxygen. What are we going to do, okay? What do you think those cells are trying to do? What are they going to do? That's a pretty bad situation, right? Because you have two things at the same time. What can those cells do? Well, yes, but that's like the, yeah, that's the end. That's the end. They will probably do it at some point, but.
Anaerobic glycolysis produce a lot of lactate, which degrades the cells and will destroy them.
Potentially, but that's absolutely what they will try to do, okay? So because they will be signaling through all those signals at the same time, one of the things that they will do is to switch to anaerobic or to glycolysis. They will try to upregulate glycolysis as much as they can. It's obvious, right? Because that's what they're doing basically in both situations. The trouble is, you're not getting any glucose or very, very little glucose, okay? Because the blood vessel is blocked. So you're lacking both nutrients and oxygen. So the only thing that the cells in the extreme situation can actually consume is their own stores, right? So if they have glycogen, they will use glycogen. If they have lipids, well, they're not very useful because we don't have enough oxygen. They can't really do very much with that. So they will use up any glycogen they may have, if they have any. And after that, they are pretty screwed, right? Because they can't really do very much. There's not enough glucose coming in. There's not enough oxygen. Yeah, they will start dying. Yeah.
Tissue degradation.
Huh? I mean, that's what will happen. So as the cells die, and either they die by apoptosis or by necrosis, depending on how they're doing. So yes, you will get basically destruction of the tissue. But the time in which the cells are trying to survive by adapting their metabolism and just doing something is actually clinically extremely important. Because nowadays in many countries, like actually in Czechia, where we have really, really good system for taking care of patients with heart attacks and also with strokes, people will get care within tens of minutes, wherever they are in the country. Okay, very quickly they will get a PCA basically, and the blood flow is restored. And it is the minutes that are between the heart attack and the care that they receive that basically decide how big a part of either the muscle or the brain will be dead. So knowing about these adaptations that the cells can do and potentially supporting them, there's a lot of research in trying to basically save as much tissue as you can, are quite important. Because it's not really, okay, blood vessel closes, everything is dead. That's not how it happens, okay? The cells will continue kind of surviving for quite some time. And yeah, it's important to know what kind of adaptation mechanisms they use, okay? So ischemia is a combination of those two things together. The last strange situation, but again very important clinically, that I want to talk about and how cells adapt to it is actually cancer. So for cancer cells, but not just cancer cells, but any cells that want to grow, the priority is to build up their components and to be able to divide, okay? It's not just cancer cells. For example, stem cells in our hair follicles or in our intestines are doing the same thing. They're just trying to build as much stuff as possible and grow as fast as possible. That's their priority. But for that, you also need to rebuild your metabolism, because the normal metabolism that we mostly talk about, you know, when we talk about metabolism, we usually think, okay, we need to produce enough ATP and maybe some components, but we're not really trying to just make as much stuff as possible. But cancer cells and some stem cells, some rapidly dividing cells are really prioritizing making as much as possible in basically the shortest period of time. So they're not so interested in making ATP really, interestingly, but they're interested in making their components. So how do cancer cells and other rapidly growing cells, how do they adapt their metabolism in order to get this priority right? So cancer, rapidly dividing cells. So one of the interesting things in both cancer cells and other types of rapidly dividing cells is that they are not really using a lot of oxidative metabolism. So when they get glucose, and many of them use a lot of glucose, especially cancer cells, but not just cancer cells, they will push most of the glucose just through glycolysis, but not really through the Krebs cycle to the complete oxidation. Now, this effect, or this observation, that mostly cancer cells, but other rapidly dividing cells as well, are producing a lot of lactate, even in the presence of oxygen. So most cells, if you put them without oxygen, they will start producing lactate. That's the only possibility, right? We just talked about that. You have to switch to anaerobic glycolysis because that's the only possibility. But many cancer cells and rapidly dividing cells will produce quite a lot of lactate and will consume massive amounts of glucose, even in the presence of oxygen, okay? This observation, this effect, using a lot of glucose and producing lactate in the presence of oxygen, is called the Warburg effect. Otto Warburg, a very important biochemist of the first half of the 20th century, and he discovered this, okay? He noticed that cancer cells are taking up massive amounts of glucose and producing lactate, even when they have enough oxygen, which is unusual because most other cells will only do it in the lack of oxygen. Now, why do cancer cells and rapidly dividing cells do this? Well, there are a lot of hypotheses, but I think the most convincing hypothesis is that rapidly growing cells need to build a lot of their components. They need to build amino acids and proteins and lipids and cholesterol and whatever. For that, they absolutely need a lot of carbon chains. Now, if you put the glucose through glycolysis and the Krebs cycle, you basically oxidize all of the carbons into carbon dioxide, right? You're left with nothing. You're just left with carbon dioxide that you breathe out. But that's not actually very useful for cells that need to build up the components. So what they do is they metabolize glucose to pyruvate, or sometimes they use even the intermediates of glycolysis, and use those carbon chains to then build lipids and amino acids, et cetera, and only some part of it will then get turned to lactate. I'll say it again. Most of the glucose, these massive amounts of the glucose that flow in, are not actually completely oxidized, because why would you do that if you need to build your own components? An interesting counterintuitive thing about cancer cells is that actually, and I already mentioned it, they don't actually need that much ATP. So the other problem, if they put all the glucose through the Krebs cycle, they would be making too much ATP. If you think about it, why would that be a problem if you start making too much ATP? Yes. And what metabolic processes are inhibited if you have too much ATP? Not gluconeogenesis. Lipid synthesis, not really, no. If you have too much ATP, there's one specific pathway which will have real problems. Possibly, but not really. One pathway that actually produces a lot of ATP, and it will really struggle. The electron transport chain. Okay? Why? The ATP synthase is synthesizing ATP. Now, if you have a lot of ATP, it will be inhibited because it can't really, remember, the free energy is really given by how far from the equilibrium you are. If you have a lot of ATP, you're moving further and further away from the equilibrium, and at some point, the enzyme will be unable to make more ATP because there's just so much ATP, okay? That inhibits the ATP synthase, and if you don't have working ATP synthase, the gradient, the proton gradient, will increase, and at some point, the electron transport chain complexes will not be able to push any more protons because there are just too many protons there, and it stops the electron transport chain. But the rapidly growing cells need the electron transport chain, for example, for the synthesis of nucleotides. We'll cover that in the next course or the course after when we talk about the synthesis of nucleotides, but there's one reaction that actually requires functioning electron transport chain. So if you have too much ATP, you can't make pyrimidines, you can't make nucleotides, therefore you can't divide. So cancer cells are actually trying to prevent having too much ATP, and that's why they're using this inefficient glycolysis pathway, because first of all, it gives them these carbon chains that they need, and second, it's not producing too much ATP that would really give them problems. Okay, so this is kind of a specific, special thing about cancer metabolism and metabolism of some other normal rapidly dividing cells, which kind of shift this thing. The final bit, and I can see I'm already a minute over, a lot of these types of things, cancer cells and rapidly dividing cells, use also as one of their major source of carbons, really, they use glutamine. How does it work? Glutamine goes in. It gets converted to glutamate by glutaminase. Yeah? Glutamate gets converted to 2-ketoglutarate by glutamate dehydrogenase. Excellent. Okay? And 2-ketoglutarate, which is of course part of the Krebs cycle, will get converted, and now this is the weird thing, will actually go the wrong way around through the Krebs cycle and will get turned to citrate. Right? Normally 2-ketoglutarate would go this way to get turned to succinate and fumarate, etc. Yeah, that's the normal way. But in cancer cells, rapidly dividing cells, it will actually go the opposite way to get turned to citrate. And why do we need citrate? What do we need citrate for? Well, yes, but here we are turning the Krebs cycle the wrong way around. For lipid synthesis, for fatty acid synthesis, the only way we can get acetyl-CoA for fatty acid synthesis is through citrate. We need to first make citrate, and then we make acetyl-CoA, and then we make malonyl-CoA, etc. Okay? So this is exactly how cancer cells and rapidly dividing cells get carbon atoms in order to make lipids. They basically switch the Krebs cycle to the opposite direction, and they use glutamine, then to citrate and to fatty acids.
Will it affect the urea cycle? Because we have also
The urea cycle only exists in the liver, possibly a little bit in the kidneys, okay? So that's a very special situation. Here we're talking about all the other types of cells, okay?
So if we will have like cancer, hepatic cancer.
Yeah, it will probably not be doing a lot of urea cycle, okay? Because urea cycle is really one of those things like gluconeogenesis, where the cells are doing it for the rest of the organism. They're not doing it for themselves, right? They're doing it to clean the rest of the organisms of ammonia, okay, or to produce glucose. In cancer doesn't care about the rest of the organism, so they will probably downregulate all those things and just push for making more nucleotides, fatty acids, proteins, just build their own cells. Make sense? Good. So those are weird situations in cells, but they are very important clinically because obviously we want to, especially for cancer, we want to understand how cancer cells are metabolizing and how we can kill them. All right, that's it for today. All right.